Today, Benjamin Crain will be presenting his research on the Palau Orchid Conservation Initiative, an integrated approach to orchid research and management. Okay, Benjamin, whenever you're ready, you can start.
All right, thank you. Okay, can everyone see that?
No, not yet.
Okay, let's try again here. Oh, there we go. Okay. My apologies. And how about now?
Nope, that looks great.
Okay, great. Excellent. All right, well, let's get started. So hi, thank you all for being here with me today, and certainly thank you for the invitation. As previously mentioned, I'm Dr. Benjamin Crain, and today I would like to talk to you about the Palau Orchid Conservation Initiative, which is a collaborative project that's based out of the North American Orchid Conservation Center here in Washington, D.C. So I'd like to start by talking a bit about who's involved in this project. As I mentioned, it's very collaborative, it's highly interdisciplinary, and we involve a team of scientists and conservation managers, really from throughout the world. As I mentioned, I'm based out of the North American Orchid Conservation Center, which is a partnership actually through the Smithsonian Institution and the U.S. Botanic Garden, among others. We also partner with folks out in Hawaii at the U.S. Forest Service and the Institute of Pacific Islands Forestry. We have a fantastic collaborator at Illinois College, Dr. Larry Zettler, who runs a lot of the mycorrhizal work that we do for this project in his orchid research lab in Illinois. And we also partner locally with several groups in Palau, including members, staff members, and researchers at the Palau National Museum in Palau, which is home to the most extensive herbarium in Palau, as well as a native plant garden. We also work with the people at the Bureau of Agriculture who are involved with the Ministry of Environment and also the Protected Areas Network in Palau. And of course, we work closely with staff and managers at the Nardoc Nature Reserve, which is where we focus a great deal of our research. It's one of the largest protected areas in the country. So as I mentioned, it's a very collaborative project and highly interdisciplinary, involving people from around the world. So given that diversity, what is it that really brings this group together? What is our overarching goal? And quite specifically, it's really to develop and implement an integrated approach to understanding orchid ecology and communities and their conservation and management in Palau. And we focus really on three kind of specific themes. First, we focus on a lot of spatial and environmental characteristics and attributes and how those focus or affect orchid communities in Palau. And then we look at things more locally in terms of vegetation attributes and forest dynamics on specific sites and determine, again, how those can influence orchid communities. And finally, we focus a great deal on symbiotic relationships and specifically orchids' relationships with mycorrhizal communities and how that affects orchid communities. And again, as I mentioned, this is an integrated approach. So we're also interested in how all of these things actually interact with each other and influence orchid communities across scales and via different mechanisms. So given that, the first question you might be asking yourselves is why orchids? Why are we interested in the orchid family other than the obvious beauty of many of its species? And there are a number of reasons. First of all, it's, dare I say, unfortunately, the world's second largest plant family. It was widely recognized as the largest until just recently, but in either case, well over 25,000 species of orchids around the planet. So it's really representative not only of plant diversity, but of biodiversity as a whole across the planet. So it's really an important group if we are considering biodiversity conservation in general. Secondly, it's distributed almost everywhere around the planet, with the exception of Antarctica. And so Orchidaceae offers a great deal in terms of potential for biogeographical studies in unique systems and across different scales, so that we're able to test some of these theories and trends that we might be observing. In addition, orchids really exhibit complex lifestyles. They range from terrestrial species to epiphytes in the treetops. Many of them have very specific pollinators. Many are dependent on microheterotrophy, which means they're dependent on a specific mycorrhizal fungus for nutrients throughout their life cycle. So they offer a great deal of potential in terms of ecological studies of plant, fungal, and animal interactions, and we can learn a great deal from those interactions and apply that to conservation, not only of orchids, but of the species that interact with them. At the same time, orchids are very sensitive, and they're very indicative of disturbances in many cases. So things like climate change, fires, and poaching can be really detrimental to orchid populations. And so they can act as canaries in the coal mine, if you will, or indicators of ecological health. And they can kind of forecast maybe potential effects of disturbances before they spread out into larger ecological communities, so that we might respond to them in a timely manner before larger effects take place. Now, unfortunately, because orchids are so sensitive, of course, many of them are known to be very rare, endemic, or endangered. And so I think they're very much in need of attention from conservation practitioners. This graphic shows a project done from the IUCN Orchid Specialist Group, and it shows the cumulative percentage of threatened orchid species that have been assessed over the past 20 years. And you'll see that the trend is kind of moving downward, but largely that's due to an increased number of assessments in recent years. And whatever the case may be, you'll still see that the overall proportion of orchids that are considered threatened around the world is well over 50%. So certainly there's cause for concern there, and they are deserving attention from conservation practitioners. So I hope I've really convinced you why I find orchids so fascinating as a research subject, but you might also be asking yourself, well, why Palau? How did this group end up working on communities in Palau? Well, aside from the obvious Micronesian charm, if you will, Palau is special for a number of reasons. And I'd like to start by just looking at a brief bit of geographical background on Palau. Looking at this map, this is where Palau is located. Here's the blow up. And the first thing you obviously notice is that it's relatively small. It's quite small. There's a total area of about 459 square kilometers, which for those of you maybe in Hawaii is about 1.5% of the area of Hawaii. In DC, where I'm at, it's about 2.5 times the size of DC for the entire country. But that said, it consists of over 344 islands that are spread over 650 kilometers in kind of this northeast to southwest direction. The largest island, Babeldab, is located here. And again, it spreads out through these many small islands towards the southwest. Now, on these different islands, we have very distinct habitats. So the larger islands here are of volcanic origin, and they have very highly weathered soils and relatively dense forests. But as we move to these smaller outlying islands, these are mostly calcareous limestone islands that were uplifted from the seafloor. So there's a very different substrate, a different history, and very unique habitats that occur on these numerous islands to the southwest as well. Now, for those of you who are students of biogeography, another thing I'd like you to notice is that Palau lies on this border between the Philippine Sea and the North Pacific Ocean, the Northwest Pacific Ocean, in this region here. And if you follow that boundary down, you pass through this region, which, for those of you who may recognize it, is delineated by what's called Wallace's Line. It was delineated by Alfred Russel Wallace a long time ago. There are numerous variations of it. But essentially what it is it's a boundary, a biogeographical boundary that separates really distinct ecological flora and faunal communities from Asia versus those of New Guinea and Australia. And so I find that very interesting, and I think for that reason alone, Palau is a very unique living laboratory and ideal for exploring things like diversity and distribution of orchid communities. Now, in addition to that, Palau is actually a very unique portion, the westernmost portion of what's delineated as the Polynesian Micronesian Global Biodiversity Hotspot. For those of you not familiar with these hotspots, what they are is very small proportions of land on our planet that are occupied by an abundance and an overabundance of overall biological diversity. So high levels of biodiversity in a very small area. Now, secondly, to be defined as a biodiversity hotspot, unfortunately, these are areas that are highly threatened and have experienced a great deal of habitat loss. In the case of the Polynesian Micronesian biodiversity hotspot, estimates show that over less than 20% of the original habitat is actually intact in these regions. Now, fortunately, Palau has some of the most intact forests in all of Micronesia, but that being said, it is not without its threats. And one of the biggest threats is forest clearing and fires in Palau, such as you see here. This graphic here, produced by some of our partners at U.S. Forest Service in Hawaii, shows wildfires on Babbledab Island, which is the largest island in Palau, over a period of about 7 years in the recent past. And just what I'd like you to notice is that they're fairly widespread, and they happen throughout these major areas of Palau. And on average, they're losing approximately 1% of their island area to fires each year. Now, in addition to threats from fires, obviously being an island nation in the Pacific, Palau is highly subject to climate change and oceanographic change. This map actually shows island susceptibility. It was produced by a group out in England. And if you look at Palau over here, not all of the islands, but several are in the high category of susceptibility to climate and oceanographic change. And so certainly this can have effects, serious effects on orchid communities there, certainly relevant to developing conservation plans. So with that, I hope I've been able to show you why I'm so interested in orchid communities in general, but specifically orchid communities in Palau. And so with that, I'd like to get to some of the primary objectives of our project and addressing those three themes that I mentioned earlier. And again, the first of which is to conduct a floristic assessment of orchids in Palau to quantify orchid diversity, their distribution, and biogeographical trends. So looking at these spatial environmental attributes that are affecting orchid communities and distribution. So for this component of the project, we started in the herbarium, and we wanted to look for historic records, if you will, of orchids in Palau. And it's worth noting that this was a really valuable endeavor. We had a number of specimens at the U.S. Herbarium here in D.C. We assessed databases really from around the world. And one of the reasons that was so critical is that many of the collections from Palau and many of the documented records were collected very early in the 1900s, particularly by a gentleman named Rudolf Schlechter. And Schlechter was from Germany, and unfortunately, his collections were completely destroyed in World War II. And so he had documented not only the diversity of orchids in Palau, but he had actually described a number of those species. And so his type specimens, his descriptions, his drawings were almost all virtually lost. So having access to these particular specimens was really critical for us getting a baseline estimate of what species occurred in Palau and where they occurred. And so currently we have about 250 herbarium records that we have been able to examine in detail, many of which had specific distribution data and information on habitat, associated host trees, things of that nature. And so that proved very valuable for us getting a baseline checklist, which contained about 80, 85 species and a general distribution of what was known to have occurred in Palau in the past. Now, of course, we weren't entirely happy with that and stopping there. So we needed to conduct field surveys to see what is currently out there. And so thus far, we've been able to conduct surveys in 11 states on both the volcanic and rock islands. And you can see here, this is one of the the employees of the Nardoc Nature Reserve getting GPS coordinates of an orchid specimen in the Nardoc Nature Reserve. Here we are again on this volcanic island, Babbeldaub. You can see the highly weathered volcanic soils here, getting samples of terrestrial orchids. And of course, we use a very different approach on these rock islands. These are smaller, more isolated islands. So we partner with a lot of local folks in Palau. This is Dr. Miles, who allows us to use his boat to go exploring these islands, Dr. McCormick at Smithsonian. And what this allows us to do is actually take advantage of the tide, as you see, and really get up into these smaller islands and access overhanging branches and actually climb some of the islands and survey some of the trees. And we can see, again, wonderful orchids in all these sites that have rarely been documented before. So we're really happy to have been able to conduct really extensive surveys in a number of distinct habitats. Some of the things that we found are really interesting. So just some background numbers. Our final list of species is up to 99 species that we've documented in Palau, and we're still counting. We're still finding new things every time we go out there. Some numbers, 38% of the species we found are terrestrial on Palau, which I feel is actually relatively high, but of course 62% are epiphytic. The majority of orchids generally are epiphytic. Really exciting is that almost 30%, 29% are indeed endemics. Same example, Crapidium palauensis, only found in the islands. But at the same time, we're also seeing that exotics are coming through here. 4% of the species we documented were indeed in exotic species, and some of them quite invasive. Some really exciting finds. We were able to document new records of orchids in Palau. This really kind of showy Cymbidium finlaysonianum. On the other hand, we also found new records like the Dendrobium criminatum, which is actually somewhat invasive in some places. I know in like the Caribbean, for example, this is kind of a species of concern, but we're not sure of its origin necessarily in Palau. We also found new varieties. This is a really nice specimen of Crapidium calcareum. It's a Palauan endemic that's only found on these rock islands, but they are these wonderful yellow flowers that only occur in a single population found on a single rock island. The rest of the species are more kind of this maroon color you see in the background of the slides. And we're even working on describing new species. This is a species of Agrostophyllum that up to this point hasn't matched anything that we know about, and so we're working on describing it hopefully very soon. So again, new occurrence records, new varieties, new species, all very exciting in terms of the orchid communities of Palau overall. Now, on the other hand, as I mentioned, a lot of these species were described a very long time ago. Communications weren't so good, and one such example is this specimen described by Rudolf Schlechter, which is Oberonia palauensis. It was listed as endemic to Palau. It had been recorded very few times. So we were fortunate enough to get out and collect some specimens of Overonia, and we were able to do some high-resolution microscopy of those flowers. And after reviewing the description that we had available without a type specimen of Schlechter's description, it seemed remarkably similar to a species called Overonia equitans, which had also been described from Palau and which was much more widespread. So using samples from the field as well as herbarium records that we had here at the Smithsonian, we actually did a DNA analysis and created this tree here. And basically what it told us was that indeed Overonia palauensis was synonymous with Overonia equitans, which had been described earlier, and therefore Overonia palauensis is reduced to synonym. So that wasn't so exciting to lose an endemic species, but from a conservation perspective, that's very important because we can now channel our efforts to species that are actually more threatened and rarer and perhaps endemic and in need of attention, rather than focusing on something that's much more widespread, not only in Palau, but in other parts of Micronesia. So as we collected our distribution data, we were also able to compare the historic records that we had from herbarium specimens to what we found in our surveys. And I'd just like to point a few things out with these maps. This is just some preliminary data. But again, this is the distribution of historically documented orchid occurrences in Palau. And here are the sites where we documented orchid populations in blue here. And what you'll notice in both maps is that the distribution of orchid communities is actually very widespread in Palau. So that's rather encouraging. It seems as though many orchid communities have remained intact over time based on what we're seeing initially here. At the same time, there are places where a number of orchid communities were documented where we aren't seeing as many, but we're also documenting orchid communities on islands where they had not been documented before. So it's encouraging to know that there's abundance of orchids here, and that an initial observation suggests that they're being fairly well protected since these historic records have been collected. Now, perhaps more importantly, this distribution data allows us to test a number of things and look at a number of trends. And I'd like to just quickly talk about a paper that was published recently from a friend of mine out in California that looked at orchid diversity in Southwest Pacific. And what they looked at was general biogeographical models. And what they found is that traditional variables like island area and isolation were indeed important for identifying and determining orchid diversity. They also found that things like climatic variables, temperature range, and topographic variables like curvature were also important. And overall, they concluded that habitat heterogeneity was really the driver of species richness in these island systems in the Southwest Pacific, places like New Caledonia, for example. So we wanted to look at how those same principles might be affecting orchid diversity and distribution in Palau. So we looked at general island biogeographic theories and just made some preliminary models based on our data. This is a species area model where you have the area of island groups, in this case, against the number of species, the log number of species on the y-axis. And what you can see by its position here is that Palau is actually performing above what we would expect based on this simple regression line here. For those of you in Hawaii, you guys have only 3 species, so you're kind of this outlier down here. Interestingly enough, maybe that's skewing the line, but again, Palau is overperforming what we might expect for its overall area, so there must be something else that's going on. Well, another island biogeographical model is the species isolation model. What this suggests, as you can see on the x-axis here, is that as an island is more isolated from its source pools of species, the immigration rate drops. So there'll be less species immigrating and colonizing and populating those islands as you move farther away from source pools. So I'll bring you back to our map of Palau's location, and you can see it is sitting right out here. So it is isolated in many ways, and that affords it opportunities for colonization and speciation to occur in isolation. But again, as I mentioned, that Wallace's line is right here, and you can see New Guinea, the Philippines, all these regions are the most immediate source pools for immigration in Palau. And so I think that that may have a real impact on its overall diversity. Well, again, when we just look at the results of our floristic surveys, we can see that that may exactly be the case. So we have species like Acreopsis liliofolia, which is widespread. It occurs in Asia, Australia, and the Pacific, including in Palau. But we also see these species that are more isolated, and they only occur in these specific islands, Palau, Guam, and the Marianas. So this likely evolved out in this isolated setting. Now, we also have directional influences, as you can see on the bottom. So, for example, we see Dendrobium mirabellianum in Palau. It also occurs throughout much of Australia and New Guinea. On the contrary, we see northern influences where we have species like Phaeus tankerville, which occurs in China, India, all the way north to Japan. So I think that because Palau is sitting on this really unique boundary, it's being disproportionately affected by these source pools that are driving its diversity. Now, in addition, I also mentioned that overall habitat heterogeneity was the big factor in driving overall orchid diversity in several other studies. And this is an example of, I think, how this is playing out in Palau. So in these rock island formations, where we see this as calcareous, limestone, uplifted, highly exposed, very sunny environments versus these volcanic islands in Palau. It's a much more shaded, dense, cooler forest, if you will, like you might expect in a typical tropical forest. And we see differences in those habitats specifically. So, for example, on the rock islands, we have both endemic terrestrials like this Crapidium calcareum here, as well as endemic or at least rock island limited epiphytes like the Sarcanthopsis. At the same time, we see that same kind of association with things like Peristylus palauensis and Dipodium freycinetioides, which only occurs terrestrials and epiphytes on the volcanic islands, and they don't spread to these other habitats. So clearly there's an association with different species and different habitats. And so given that all together, these are the types of things that we're going to need to take into consideration as conservation practitioners if we're looking at protecting and restoring not only the most diverse areas, but also recovering areas throughout the different habitats of Palau. Okay, so now that we've looked a bit about some of the spatial and environmental characteristics that are controlling orchid diversity in communities in Palau, I'd like to switch on to that second topic, which is specifically to determine how local vegetation and habitat attributes influence and shape orchid communities in Palau. And for this part of the project, we focused a lot of our research on the Nardoc Nature Reserve, which is located in Melekeok, Palau, and the largest island of Babbledog, as you can see here. It's the largest protected area in the country, and it was basically protected to protect a freshwater lake, as well as the surrounding forest. But it has had a history of land use and land use change throughout parts of it, as well as protecting several really old, mature forests. Perhaps more importantly, the Nardoc Nature Reserve is also home to the most recent Smithsonian Forest Geo Network plot. For those of you who are not familiar with this is a series of plots that have been established throughout the planet. And basically, they are permanent plots that are continuously monitored that are available for people to conduct long-term research on these forest systems. They're all done systematically. So on each plot, all stems greater than 1 centimeter in diameter for all the trees are ID'd, measured, tagged, and mapped, and then they are remeasured through time for long-term studies. So working with partners in Hawaii that manage the Nardoc Nature Reserve forest plot, they have been able to document over 13,000 trees that have been mapped, ID'd, and database to this point. And this is a graphic of the Nardoc Nature Reserve plot here. You can kind of see in this northwestern and southeastern range, these are the more mature forested areas. This is a site that had been previously used for agriculture, and now it's in several stages of recovery as you cross these gradients. But you'll notice the differences in tree species in these different areas, and those have actually equated to the identification of 6 distinct habitat types that our partners have been able to identify and quantify on the site. So you can see here, for example, these are these badland type regions that are basically just essentially bare rock, moving into these fernland areas and forest edge, savanna areas. And then you can see these more mature forests here in the background as we transition away from these disturbed sites. So for looking at the orchid communities and how they might be affected by all these differences in the local habitat conditions, we conducted orchid surveys on 99 subquadrants, which is 1 subquadrant in each quadrant of this forest geoplot, by walking transects and at 1 meter intervals, and documented each individual orchid that we encountered. Now, when we encountered an orchid, we first identified the orchid itself, of course, the species, and then we identified each of its nearest neighboring trees in the 4 cardinal directions. In addition, for epiphytic species, we noted its position in the host tree itself in terms of cardinal direction. We also noted its position in each of 5 canopy zones, and obviously identified the host tree itself. So in doing so, this is the map we've been able to produce thus far. We've actually mapped over 5,000 individual orchids on the entire plot itself. This map in particular shows the distribution of terrestrial orchids on the plot. And again, what you'll see is that there are distinct differences between these more mature forested areas in the northwest and southeast, and then these disturbed sites that are centrally located. The colored dots represent different species. You can see differences in terms of abundance. And so in looking at these communities, specifically, for example, we're in preliminary stages of analyzing this data, but we did see distinct differences in terms of orchids themselves. So on the savanna sites and the more exposed sites, we're seeing native things like Spathoglottis carolinensis, which is native, however, it's disturbance prone. But also these exotic species like the bamboo orchid seen here, which is actually a more invasive species. However, moving into the more mature forests, we start to see great abundance of these endemic terrestrials like these Crapidium pristinianum and Crapidium satypes, which are both really never found outside of these more mature habitats. In addition, again, we haven't analyzed these data yet, but we have made observations that indicate certainly mature trees tend to house greater abundance of orchids, as you can see, for example, on this tree here. There seem to be species preferences. I believe this is a Campnosperma. We seem to always see orchids on those particular species. However, things like pandanus and some palms, we just don't see things on them so much. So there certainly seems to be preferential favoritism towards certain species of trees by orchid communities. And we're also seeing that in terms of canopy structure. For example, the Oberonias that we're finding are almost always branch epiphytes, more in exposed areas of the crown. But we also see things that are low trunk epiphytes, like this Dendrobium brachyanthum, which doesn't get much above head height in any instance. It's really locked into that lower, cooler part of the canopy itself. So again, from a conservation perspective, this is all important information for when we're considering recovery and restoration efforts. Certainly different species are going to be more appropriately situated in different habitats, not only in the overall habitats themselves, but within the tree structure itself. And so that's an important consideration that will help guide reintroduction decisions when the time comes. Okay, so now that we've looked at, again, these broader spatial patterns, but also attributes of local vegetation and how that affects orchid communities, I'd like to finish by looking at how relationships with mycorrhizal fungi regulate orchid diversity and distribution patterns. Now, so for those of you who aren't familiar with this association, the association between orchids and mycorrhizal fungi is a necessary symbiosis, and it starts at germination. So an orchid seed basically has no nutrient tissue. Okay, so as I was saying, I'll back up just a second. I was mentioning that orchids and mycorrhizals are a necessary symbiosis. It starts at germination. So orchid seeds have no nutritive supplies that allow them to germinate. So they require colonization of these mycorrhizal fungi that you see here in order to germinate and grow until they can start producing their own food via photosynthesis. Now, at that point, the relationship doesn't end. And so this diagram shows a cross-section of a root here. And this is orchid mycorrhiza that eventually penetrates those root cortical cells and forms what are called pelotons that you can see here. Here's an image of it. Here's an image of it here. So these are nutrient filled, and as the orchid, as it grows, needs those nutrients, it will begin to digest those pelotons and use them as needed. Now, it's interesting to point out, and this is an important point, that several orchid species are very specific, and they require only a specific fungi to germinate. Others are more general, and we're interested in how that might affect distribution, abundance, and overall orchid communities overall. So To look at this in Palau, we sampled root samples in the field. We're up to about 500 samples at this point, and we sample them in a very specific manner. So we look for living, healthy orchid roots that are actively growing, and we look for roots that are either submerged in the soil or oppressed to a bark substrate in the case of epiphytes. And what that means is that they're in direct contact with their substrate, and so they're, the potential orchid mycorrhizal fungi that are on that substrate are available to that root specifically. Now, when we collect the roots, we take half of them and we store them immediately in CTAB buffer, which is a buffer that's used to preserve DNA. And we can extract that DNA not only from the plant, but from the fungus inside of it back at the lab later on. Now, in addition, we take the other half of our roots, and we store those in a sterilized, humidified vial. And essentially, the aim of that is to keep the root tissue and the fungal tissue living inside it alive until we can get it back to process it in the lab as well. Now, while we do that, we also take samples of the substrate. And what this allows us to do is look at the potential mycorrhizal fungi that are available in an orchid-specific habitat. And that gives us an idea of whether or not something might be using a specific fungus because it's the only thing there, or if it's using something because it's very specific to that fungus. And that all depends on what's available to it. So by sampling the substrate, we're able to assess what's in the soil, how abundant it is, and which of those potential associates the orchids themselves are using. So, as I mentioned, our first samples that we collect that are preserved for DNA analysis, we take back to the Smithsonian Environmental Research Center and process them. They're then sent to the Natural History Museum and the Laboratory of Analytical Biology, where they are sequenced to produce DNA barcodes. These DNA barcodes allow us to identify the different orchid mycorrhizal species. We're able to upload these into GenBank, which is basically a database of all the species, or what are called operational taxonomic units when they haven't been fully identified, but they are distinct species, if you will. We're able to compare them to what's known in GenBank and also identify things that have not been identified previously. And what this also allows us to do is look at which specific fungi are the orchids of Palau associated with it, and how are those associations affecting things like distribution and abundance. And so this graphic shows in detail what we actually found up to this point. And so essentially what we found is that specificity, things that rely on a specific fungus, generally tend to be the endemic species. And let me point out a few things. So on the x-axis here, we have individual orchid species. And on the y-axis, we have the percentage of roots that we sampled that had the dominant orchid mycorrhizal fungi associated with that species. So in other words, we find everything that's associated with that species, and we identify the one that's most commonly associated with this. And what that tells us is that this is the most important OTU that the orchid is reliant upon for germination and survival. And if we look at our different groups, we can see our endemic species in the green and our widespread species in the red, or I'm sorry, in the blue. What we see is that there was a significant difference here. So the widespread species tended to rarely associate with a single dominant mycorrhizal fungi. The endemic species regularly tended to associate with not only a single one in many cases, but regularly almost always. So that was a distinct difference based on an in-orbit test. We also compared epiphytic species that you can see in these darkened bars to terrestrials. There was no difference there. And as you can see, we looked at abundant species versus rare species, and there was no difference there. So again, in terms of specificity, endemics tended to be more specific, and therefore that's likely a driver of why they're endemic to Palau, is that they could only use a single fungus in order to persist. Now, as I mentioned, we also took these substrate samples to see what was available to our orchid communities. And this graphic shows individual mycorrhizal fungal OTUs, or individual species, if you will, And on the y-axis, it shows the number of sites in which that species was found. Now, the green bars here delineate those mycorrhizal fungi that were associated with orchids themselves. And what you'll see on this graph is that basically the majority of orchids associated with mycorrhizal fungi that were found at a great deal of sites. The fungus that was not found very frequently, rarely, but not necessarily never, was associated with orchid species. And so we looked into that and how that affected our different groupings. And what we saw was something relatively different. And so if you look at our endemic species in the green here versus our widespread species, there was really no difference in terms of the percent of sites that had their appropriate mycorrhizal fungi. So fungal abundance wasn't really affecting endemism. However, when we compared abundant species and rare species, whether they were endemic or whether they were widespread, in other words, occurring outside of Palau, there was a distinct difference. So really, mycorrhizal fungi abundance as far as its distribution in Palau really affected the abundance of individual species that associated with those fungi, whether or not they were endemic to Palau or occurred elsewhere. And so from a conservation perspective, again, this is really important because not only do we now know which species of orchids associate with specific fungi, but we also know that some are very much more specific and they associate with fungi that are far rarer than other more common species that might be out there in these habitats. So the next question really is then how can we equate that directly into orchid conservation? And this is where our living roots come in that we sampled. And so we take those living roots back to an orchid research lab with a fungal isolation quarantine lab at Illinois College, Dr. Zettler's lab. And we process them to actually release those living pelotons under sterile conditions. And to do so, we chop them up in a sterile hood. They are then cultured in a fungal isolation media, which is a media that's specially designed to slowly grow these pelotons. And in doing so, we are able to isolate specific hyphae and specific pelotons so that we know we have a pure culture that we can work with. Now, thus far, the results of these efforts have produced 63 different fungi from 17 species of orchids that have been cultured, pure cultures. They've been ID'd. We take samples of them. We analyze the DNA, and again, we barcode those and put those on GenBank. And they're also preserved at the University of Alberta Microfungal Herbarium, where they're cryogenically preserved so that we can resample those at later dates as needed again for genetic analysis and for other uses. So that's been a really great partnership so far. And most importantly, those living cultures we can then use to test symbiotic germination trials. So what I mean by that is that there are a number of fungal partners that can actually exist in orchids and orchid roots that aren't necessarily suited to germination. So we isolate those fungi and we have to test their mycorrhizal potential. So during our surveys, we've collected seeds from a number of different orchid species. Here's an example of one we've dissected from Dendrobium brachyanthum, which is an endemic in Palau. So we have seeds for about 25 species. You can see them here, just thousands of them. And what we do with these is we sterilize them so that there are no contaminants or outlying fungi that we might introduce in the seed coat. They're then swabbed with a bacterial swab onto a culture plate. This actually contains an agar media that's either asymbiotically useful for growing the orchids themselves, or it is a symbiotic media which is suited to our cultures of the fungi that we've isolated from that particular species roots. And we either grow the seeds asymbiotically or symbiotically and wait for our seeds to germinate, either by the nutrients in the agar or by the nutrients provided by our mycorrhizal fungi. And again, we're in the preliminary stages here, but we've had some early successes, and thus far we've actually had 5 species, including endemics like the Dendrobium crameri from Palau that have been germinated in the lab in sterile culture, and these seedlings have now grown and they are being ready, or they are now ready for being transplanted. And their final destinations involve ex situ and in situ conservation strategies. So we have a number of seedlings that are already been established and that are on their way at the U.S. Botanic Garden, and we'll be going to partner institutions. But perhaps more importantly, the majority of these seedlings, their destination ultimately is for places like the Nardoc Nature Reserve and the other protected area network restoration sites where they're actively reforesting and replanting these areas. And our hope is to be able to bring these seedlings back and to really bolster the orchid communities in these recovering forest sites. So with that, I'd like to just get to the take-home message, which is really, I hope I've been able to show you that there are multiple environmental and ecological factors that cross different scales and that influence orchid communities in various ways, and that integrated approaches and by looking at the connections between all of these things are really going to be necessary to assure their conservation over the long term. Now, in addition to that, I think that success in these projects can really only happen with not only interdisciplinary research, but more importantly, local engagement and capacity building to generate support for these conservation strategies and interest and support for the orchid communities themselves. And so we take a really active and aggressive approach to work, for example, with high school students in Palau, showing them how to create permanent herbarium records and make plant collections in the wild and do orchid identification. But we also take alternative approaches like you can see here. This is our most recent orchid gami model, which is kind of an artistic craft project that we've produced, and we've disseminated thousands of these to local community groups at ecotourism sites, at the tourism boards, in the museum itself where student groups come in, community activities, and people have really given us positive feedback and enjoyed engaging in this just really fun activity that connects them to the orchids of Palau. And you can see this is Cholet. She's one of the museum workers at the National Museum, standing in front of the plants of Palau exhibit with that particular species. So we're really excited about that, and I truly believe that it's very, very important component of this project. And so with that, I'd like to thank everybody for their time, apologize for the brief interruption, and take any questions that you might have.
Thank you so much. That was an awesome presentation, and I learned so much. Does anybody have any questions? Feel free to put them into the chat, or you can unmute yourself. Oh, we do have one question from Stephen Jones. Is it possible to study the distribution and health of the fungal communities that orchids associate with?
Okay. Yes. So I'm sorry, could you repeat that? The distribution and health?
Yes, of the fungal communities that orchids associate with.
Well, the second part of that, health, would be probably challenging in the sense that we don't really know what's out there. And I only mention that because the first part is almost as challenging. So the answer to the question is yes, you can look at the distribution. However, it's very, very difficult because there have been studies, and we're looking into how we can do this in the future, but there have been studies that show that the differences in the mycorrhizal communities, even 1 foot, a few inches apart, can be very, very different. So people have surveyed individual trees and done swabs at 1 foot intervals up a single tree, and they've got completely different results with each sample. So that may not always be the case, but I think that's one of the things that's been very challenging, and it's actually inhibited people from really engaging in those types of tests, because it would be extremely time consuming, and you would have to be extremely careful, and it would take a lot of processing. But with the way things and technology is improving and how we're able to process these samples faster and more efficiently, Those days are certainly coming, and that's a very interesting question, because when you're talking about a different mycorrhizal community from one tree to the next, and perhaps even within a tree, that can have huge implications on where you might want to introduce cultivated species. For example, if we're propagating these endemic orchids and we're trying to reintroduce them to a recovering forest, if your mycorrhizal community can be different, in a 1 foot space, it'll be really important to make sure you're considering that. But again, tests allow us things like metabarcoding to sample different sites and determine what's there. And if you can use the barcodes that we've made available, for example, and know that this is the fungus you're looking for, you could potentially collect a large number of substrate samples and specifically identify sites that have what you're looking for. So the potential's there, but very, very challenging, very challenging.
Okay, awesome. Thank you. Makuni Greg says mahalo. That was awesome. I also have a question. I'm not sure if you mentioned this, but how do the orchids normally get their mycorrhizal fungus? Is it mostly from like their parent plant?
Actually, that's a great question. That's a very interesting question. And the way you worded it, I don't believe, well, I don't want to go so far as to say that they may be able to get some from their parent plant on the seed itself, but I think that's very rare. Generally speaking, it's a matter of chance. And if you look back at that image that I showed of the dissected fruit with those seeds in there, they just look like dust. And that's actually the strategy. And that's why they need the mycorrhizal fungi. The parent plant provides no nutrients to its offspring. I like to call them the world's worst parents. They provide nothing. But they have 10,000 propagules per fruit potentially. So they rely on the wind to spread as far and as fast as they can. And they're basically gambling on chance. They're basically saying, if I can put 10,000 of these out there, the likelihood that it finds the fungus it needs, that's what they're hoping for. And so it's an important question too, because that goes back to what we're looking at in terms of specificity and the distribution of potential mycorrhizal associates, because if you're a generalist, like we saw in some of those widespread species, they can basically land anywhere and they're going to find something they like. Whereas the ones that are more specific, if they go too far from their localized habitat of the previous generation, they might risk not finding that fungus. And so that is part of the reason that could explain why, again, they are endemic and restricted to a specific range. And in terms of overall abundance, orchids that you encounter frequently versus those that we see, even though they're widespread, you don't see many of them. Again, that equates to the abundance and the distribution of those mycorrhizal associates as well. And so if you're a fungus that's found commonly throughout the potential habitat, that's going to affect abundance. And so from an orchid parent's perspective, they're banking on that.
Okay, awesome. I want to respect everyone's time. It is 1 o'clock. We do have another question, but if anybody has to leave right now, you can feel free to do so. Thank you so much for coming. We can, if you still have time, Benjamin, we can ask this next question.
Oh yeah, no problem at all.
Okay,
happy
awesome.
to stay.
The next question is, are the right fungi for specific orchids also associated with other plants typically that might help locate suitable sites?
That's a very open question, a wonderful question, very intriguing, and it depends. There are different groups of generally what are called OMF, or orchid mycorrhizal fungi. They're usually Ceratobasidium, Tulisnella, and the Serendipita is a more recently identified grouping that associates with orchids generally. But I'm of the hesitant opinion to limit it to that. I think that a lot of people have assumed nothing else will work, and so some things might get overlooked. But overall, most of the orchid mycorrhizal fungi that we see comes from one of those three groups. Now, the reason I mention that is because those groups are very different from each other, and there are distinct habits that they have in and of themselves. So some of those are actually ectomycorrhizal fungi with other tree species that will associate with the orchids as well. Others are just detritivores. They live on dead, decaying leaf litter or logs, and so they behave very differently, and they're not necessarily associated with a specific plant or a specific host tree as an ectomycorrhizal fungi might be. And so, yeah, so it's a good question because they're both, and it depends again on the individual orchid, the different regions you're in. These patterns change whether you're in tropical forests, whether you're in islands, whether you're in temperate forests. So again, it's really this open pool of questions that really needs answering, and all of these things are very applicable to how you might operate in terms of a conservation plan, a management strategy. It's very dependent, and so there's a lot to be learned, and that's really what excites me about all this.
Awesome. Thank you so much. That was awesome. And that, I think, is all the time we have today. Thank you, everybody, for coming. I will be sending you guys a follow-up email with the feedback form, and hopefully I will have the YouTube link up by the end of this week. And I will also be able to give you guys, Benjamin said I could give you guys his email just in case you think of any other questions. But thank you so much for coming.
Thank you. Thank you for having me.
Yeah, and everybody in the chat is saying thank you as well.
Wonderful.